Hello students, today we will discuss the first life process in detail, which is nutrition. But before we start with the topic, let us understand the meaning of some important terms. Nutrition. What is nutrition? The process by which an organism takes food and utilizes it is called as nutrition. And what is the need for nutrition? Organisms need to fulfill their carbon and energy requirements. This energy is used to perform activities, and the energy is supplied by nutrients. Organisms also need raw materials for growth and repair, and these raw materials are also provided by nutrients. Hence, there's a need for nutrition. And what is this nutrient? A nutrient is a substance which is used by an organism to survive, grow, and reproduce. The requirement for dietary nutrient intake applies to animals, plants, fungi, etc. So let us now understand how do living things get their food, or how do they obtain their nutrition. There are two modes of nutrition. Number 1, autotrophic nutrition, and number 2, heterotrophic nutrition. Let us discuss the first mode of nutrition, that is autotrophic nutrition, in detail. So what is autotrophic nutrition? The word auto means self and troph means nourishment or food. So it is that mode of nutrition in which an organism prepares its own food by using simple inorganic raw materials. For example, green plants and blue-green algae make their own food, and they use inorganic raw materials like water and carbon dioxide to prepare complex organic compounds like the carbohydrates. So all these organisms are carrying out autotrophic nutrition, and these organisms like green plants and blue-green algae which are involved in this process are called as autotrophs. So what is an autotroph? An organism which is capable of making its own food from inorganic substances like carbon dioxide, water, using light or chemical energy is called as an autotroph. For example, plants, bacteria and blue-green algae. So plants are called as autotrophs. Autotrophic nutrition is thus a way to fulfill the carbon and energy requirements of plants, which is further accomplished by the process of photosynthesis. Let us have a look at this diagram, a very simple diagram demonstrating photosynthesis. We can see here that there is a green plant which takes up carbon dioxide from the air, water from the soil, and in presence of sunlight, which is giving it the light energy, and in the presence of chlorophyll, it forms glucose, and oxygen is also released into the atmosphere. Let us understand this diagram more in a very detailed diagram of this slide. So here we see there's a green plant or a green leaf, as you can see. It takes up the water which was absorbed from the roots. It takes up carbon dioxide, the second raw material, which enters the stomata of the leaves. And the third, it traps the light energy coming from the sun to make food. The food is in the form of sugar, which is utilized by the plant for meeting its requirements. And if the plant does not need this sugar, it gets converted into starch, which is stored food in other parts of the plant. And if you test it with the help of iodine solution, you will know that that portion will turn blue-black, which we will study in our upcoming experiment. Along with sugar, oxygen is also given off in the air. If we try to look at this process in equation form, then we can see, as is depicted in the slide, carbon dioxide plus water in the presence of light energy and chlorophyll gives out carbohydrates plus oxygen, or CO2 plus H2O in presence of light energy and chlorophyll gives C6H12O6, which is your glucose, plus O2, which is oxygen. Hence, we can now define photosynthesis. So photosynthesis is the process by which autotrophs take in carbon dioxide and water, which is converted into carbohydrates in the presence of sunlight and chlorophyll. But what happens to the carbohydrates formed by the plants? As explained to you earlier, carbohydrates are utilized for providing energy to the plant to carry out its activities. But the carbohydrates which are not used immediately are stored in the form of starch, which serves as the internal energy reserve to be used as and when required by the plant. You may recall that in animals, the food which is not used immediately is stored in the form of glycogen. Let us now see what actually happens during the process of photosynthesis. So the first step is the absorption of light energy by chlorophyll. Second step is conversion of that light energy to chemical energy. The chemical energy we mean the glucose, and splitting of water molecules into hydrogen and oxygen. That is why oxygen gets released into the atmosphere. And the third step is a reduction of carbon dioxide to carbohydrates or the glucose. So these steps need not take place one after the other immediately. For example, in case of desert plants, they take up carbon dioxide at night and prepare an intermediate, which is acted upon by the energy absorbed by the chlorophyll during the day. Now, let us see how each of the components of the above reaction, which was shown to you earlier, are necessary for photosynthesis through the given activities. So first activity would be to demonstrate that chlorophyll is necessary for photosynthesis, for which we will adopt the following method. Take a potted plant with variegated leaves, for example, money plant or crotons. By variegated leaf, just have a look at the slide. On the corner right side, I have shown you a variegated leaf, which means that the chlorophyll or the green portion is not spread uniformly. It is only at few patches. Now keep such a plant in a dark room for 3 days so that all the starch gets used up. Starch is a stored food. So when you're not exposing the plant to sunlight, it will use up the stored part. So the stored food is then used up. Now keep the plant in sunlight for about 6 hours. So when we're keeping the plant now in sunlight, it should be able to do the process of photosynthesis. Now pluck a leaf from such a plant and mark the green areas in it and trace them on a sheet of paper. Dip this leaf in boiling water for a few minutes. I will explain you the importance of boiling. Why did we do it? After this, immerse it in a beaker containing alcohol. We will also see why alcohol was put in the beaker. And carefully place the above beaker in a water bath and heat till the alcohol begins to boil. What happens to the color of leaf? And what is the color of solution? We'll see it in the upcoming diagram. Now dip the leaf in a dilute solution of iodine for a few minutes and take out the leaf and rinse off the iodine solution, and then observe the color of the leaf and compare this with the tracing of the leaf done in the beginning. What can you conclude about the presence of starch in various areas of the leaf? Let us understand this whole procedure once again through this diagrammatic depiction. Here, so when we expose the sunlight, the leaf for 6 hours, after that we boil that leaf in water to break down its cell walls. Once the cell walls are broken, now we will heat the leaf in alcohol to remove the chlorophyll. And in the third step, the leaf was heated in water to soften it. And the fourth step, we put drops of iodine solution to test for the presence of starch and see what we obtain. We saw that in the variegated leaf, at the corners, there were green patches, which represents chlorophyll. So because chlorophyll was there, the food was able to prepare food only in those areas, that is the margins or the borders of the leaf. And because the leaf prepared the food, the starch would also be present only in the corners. So the iodine solution will turn only the corners of the leaf blue-black because the starch will be present only in those areas. Hence, we conclude that chlorophyll is necessary for photosynthesis. The green patches represent that chlorophyll was present there, and the pale yellow patch shows that there was a lacking of chlorophyll on that side. Similarly, non-chlorophyllous patches remain brown with iodine solution, whereas chlorophyllous patches turn blue-black with iodine solution. So this shows that chlorophyll is necessary for photosynthesis. In class 9, students, you had read about stomata, which are tiny pores present on the surface of the leaves. Massive amounts of gaseous exchange takes place in the leaves through these pores for the purpose of photosynthesis. But it is important to note here that exchange of gases occurs across the surface of stems, roots and leaves as well. Since large amounts of water can also be lost through these stomata, the plant closes these pores when it does not need carbon dioxide for photosynthesis. So, the opening and closing of the pore is a function of the guard cells. And the guard cells swell when water flows into them, causing the stomatal pore to open. Similarly, the pore closes if the guard cells shrink. Let us understand this further through the diagram. As you can see in the diagram, there is a stomata. The white color portion represents the pore called stomata and is surrounded by the green colored guard cells. The green color is because of the presence of chlorophyll or chloroplasts. So, when the cells become turgid, the stomata open, and when the cells become flaccid, the stomata close. And why do the cells become turgid? Because water enters inside through the guard cells. And why do the cells become flaccid? Because the water exits. So what controls the entry and exit of water? So look at the portion B, which says role of potassium in stomatal opening and closing. So this means that whenever the solute potassium has a higher concentration, the water will flow inside through the guard cells and cause the cells to open and stomata to open. Similarly, if the potassium concentration is low, the water will flow out from the guard cells and cause the stomata to close. We can further understand this through another diagram which clearly shows you the stomata and the guard cells. The water is coming inside because the solute potential is high. So it moves into the guard cells and makes the stomata open. In the second case, the solute potential, that is potassium potential, is low. So the water moves out of the guard cells and makes the stomata close. We have discussed about several raw materials required for photosynthesis. Now we will again see an activity to demonstrate the role of carbon dioxide, which is also a raw material during the process of photosynthesis. For this purpose, let us take two healthy potted plants which are nearly the same size. Keep them in a dark room for 3 days. We are keeping them in a dark room so that there is no photosynthesis. Now, place each plant on separate glass plates and place a watch glass containing potassium hydroxide by the side of one of the plants. Students, note here, the potassium hydroxide is used to absorb carbon dioxide. So, in the first setup, the plant will be able to absorb carbon dioxide, while in the second setup, the plant will not be able to absorb carbon dioxide because we will cover both plants with separate bell jars. Use Vaseline to seal the bottom of the jars to the glass plates so that the setup is airtight, and keep the plants in sunlight for about 2 hours. So as you can see in the diagram, you will see that the experimental setup in first case has potassium hydroxide because the watch glass contains potassium hydroxide. In the second setup, there is no watch glass, so there is no potassium hydroxide. So we will observe that in the first setup with potassium hydroxide, the amount of starch will be more and it will be produced over here in more amount as compared to the setup B. So this shows that carbon dioxide, which is also a raw material during the process of photosynthesis, is actually used in the process of photosynthesis. So far, we know that the different raw materials used by plants are number 1, carbon dioxide, which it gets from air, water, which is taken up from the soil by the roots in terrestrial plants. Other materials like nitrogen, phosphorus, iron and magnesium are taken up from the soil. Nitrogen is an essential element which is used in the synthesis of proteins and other compounds. This is taken up in the form of inorganic nitrates or nitrites, or it is taken up as organic compounds which have been prepared by bacteria from atmospheric nitrogen. The roots of leguminous plants can absorb nitrate or nitrite compounds from the soil, and Rhizobium bacteria helps in converting atmospheric nitrogen into organic nitrates or nitrites. In the next slide, you can see the cross section of a leaf. As you can see, the central main vein is called as the midrib, the flat surface represented by a lamina or leaf blade, and a network of veins. If we see this diagram in detail, we can see that the upper layer or the upper epidermis is covered with a cuticle. Cuticle is a waxy layer which helps to prevent transpiration. And in the subsequent layer, we see mesophyll, which comprises of pellucid layer and spongy layer. The chloroplasts are present in this layer. In the lower epidermis, we can see the guard cells, which help in the closing and opening of stomata. With this, we come to the end of the topic autotrophic nutrition, which was happening in green plants. But what about non-green organisms? What happens there? So, in such kind of organisms, we have seen that the organisms, they have adopted another mode of nutrition which is called as a heterotrophic nutrition. So, the mode of nutrition in which an organism takes food from another organism is called heterotrophic nutrition. So, what is a heterotroph? So, the organism which cannot produce its own food, instead taking nutrition from other sources of organic carbon, mainly plant or animal matter, is called a heterotroph. These organisms use biocatalysts about which you will learn later, which are called as enzymes. Examples of heterotrophs are humans, and other examples include fungi and parasites like tapeworm, roundworm, cuscuta, etc. Heterotrophic nutrition is classified into 3 types: saprophytic, parasitic and holozoic. So first we will discuss about the saprophytic mode of nutrition, which happens in non-green plants. Students, you may recall, green plants show autotrophic nutrition. Organisms obtaining nutrients from dead and decaying matter are called saprophytes, and their mode of nutrition is called saprophytic nutrition. These saprophytes release digestive enzymes to break down complex organic matter into simpler ones outside their body. So this kind of digestion is happening outside the body, as contrast to the human beings wherein the digestion takes place inside the body. So in saprophytes, once the food is digested outside the body, it is then ingested by the organism and utilized. So example of this kind of nutrition is fungi and bacteria. As you can see in the other slide, we are showing here agaricus, which is a fungus, and this shows saprophytic nutrition. The second type of heterotrophic nutrition is parasitic nutrition. If an organism depends on another living organism for nutrition, its mode of nutrition is called parasitic nutrition. Such organisms are called parasites, and the organisms from which they obtain nutrition is called host. The host organism is harmed by the parasite. So parasitic nutrition is observed in some bacteria, fungi, flowering plants like cuscuta and animals like ascaris. Let us see the pictures of parasites. If I talk about plant parasites, we have cuscuta or the amerbale. If I talk about animal parasites, I can see ticks, tapeworms, roundworms, lice, leeches. So all these are examples of parasites. The third type of nutrition is Holozoic nutrition. Holozoic nutrition involves intake of parts of plants or animals or an organism as a whole by the process of ingestion, which is then digested and absorbed. Some examples are human beings, amoeba, frog, insects. So we have now covered autotrophic nutrition and various types of heterotrophic nutrition. So let us now see the differences between autotrophic nutrition and heterotrophic nutrition. In autotrophic nutrition, the organisms prepare their own food from inorganic raw materials, whereas in heterotrophic nutrition, the organisms obtain nutrition from the food prepared by autotrophs or other organic resources. The processes involved in autotrophic nutrition are mainly photosynthesis and chemosynthesis, and the major processes involved are in heterotrophic nutrition saprophytic, parasitic, holozoic. Chlorophyll is present in majority of organisms, whereas chlorophyll is absent in organisms using this mode. The organisms using the mode of autotrophic nutrition are called producers, while the organisms using the mode of nutrition that is heterotrophic are called consumers. So students, by the end of this video, we have learned today the differences among the terms nutrition, nutrient, importance of nutrition, modes of nutrition that is autotrophic and heterotrophic. We have been able to define the terms autotrophs and heterotrophs, different types of heterotrophic nutrition along with examples, the process of photosynthesis, activities to demonstrate how different raw materials are required for photosynthesis with respect to chlorophyll and carbon dioxide, importance of raw materials like nitrogen, potassium, phosphorus in photosynthesis, mechanism of opening and closing of stomata. So now this is time for your question answer session. So I hope you understood the video. Otherwise you can rerun the video, learn from it, and then answer the following questions which I'm going to just now announce and write them in your notebooks. So first question would be define nutrient, nutrition and diet. Second question would be where do plants get each of the raw materials required for photosynthesis? Question number third, what are the differences between autotrophic nutrition and heterotrophic nutrition? Thank you students. I hope you understood and enjoyed the video.